In our yesterday's class, we were discussing about the locomotion and movement. So, what are different types of movements are there? So, I meant to say that in case of human beings or in case of animals, the movement of legs. I mean to say the person who is going to run, he is going to walk, or he is going to lift the hand, and opening and closing of eyelids. So, opening and closing of mouth. These all are considered as a kind of the locomotion or the movement. And all locomotions are movements, but not all movements are locomotions, as we know that. And also, the amoeba can move from one place to the another place because of to capture the respective prey is also considered as one of the movement that is called as by means of pseudopodia. And that amoeba which is going to use the pseudopodia as its locomotory organ. I mean to say for the purpose of movement. And one such organism called as Paramecium. So that Paramecium is also going to use some of the fine hair-like structures called as cilia. So those cilia are nothing but again locomotory organs. So with the help of that cilia, they can move from one location to the another location, considered as a locomotion and movement. And also a protoplasmic streaming. I mean within the cell, all the cell organelles are going to stream, or they can wander around, within the cytoplasm as well as the nucleoplasm, called as protoplasm. And there are many types, different types of locomotions, movements are there. One is flagellar movement, ciliated movement or ciliary movement, and last one is muscular movement. Again in the muscular movement, one is skeletal movement, a smoother muscle movement, and last one is cardiac movement or cardiac muscle movement. And today we are moving on to the very important topic, that is muscles. So what mean by muscle, how they are manufactured, how they are formed, yes, sir, what is the actual structure of muscles, we'll discuss in today's class. See, what are muscles? These are the very important, I mean to say almost say 40 to 50% of our body total weight is made of muscles. So when you take a muscle as a part, say for example when we have a muscle, imagine we have a muscle. So particularly when I take this as a muscle, this muscle is made up of that particular, the whole part considered as a muscle, that particular whole muscle is made up of small compartments are called as muscle bundles. Remember the word. So muscle is made up of, I mean to say the structural units called as muscle bundles. See, again when you take muscle bundles, so those muscle bundles are again made up of, they called as muscle fibers. Remember. So first one is muscle. Muscle is made up of muscle bundles, and muscle bundles are made up of muscle fibers. And the thing is, when we have a muscle, in the muscle they are made up of muscle bundles. Say for example, here we have one muscle bundle, very close to this one we have one more muscle bundle, and again we have one more muscle bundle, yes sir. So this whole muscle bundle, first like this, is the whole considered as one whole muscle, in that each and every one are considered as muscle bundles. Clear? Muscle bundles. See, in that one muscle bundle is going to attach with the next muscle bundle. Remember, one muscle bundle attached to the very next muscle bundle with the help of fasciola. Remember. So that is nothing but a kind of connective tissue. So nothing but that is simply considered as fasciola, and in that the one is considered as fasci. Just remember, fasci and fasciola are fascicles. Better word is fascicles. This is what? The muscle bundle. In that each and every muscle bundle again made up of muscle fibers like this. As you can understand, that each and every one are considered as muscle fibers. They are the structural units. Next, when you take muscle fibers, again these muscle fibers are made up of structural units called as myofilament. Yes, they are considered as muscle filament or myofilament, also called as myofibrils. Yes. So just remember muscle bundle to muscle fibers are made up of myofilament or myofibrils. So these myofibrils are the structural and functional unit of the muscles. This is what the arrangement of muscle or the muscles are made of. Next, at the same time, there are some important words. These myofibrils are the myofibrils. I mean to say the structural unit of the muscle. They are covered by, they are enclosed by, that is sarcolemma. So the word is sarcolemma. So I can compare this sarcolemma with the cell membrane. See, we can take a typical cell. That a typical cell is covered by plasma membrane, or we can call it as cell membrane. Likewise, in myofibril or in the myofilament, they are covered by, they are surrounded by the membrane called as sarcolemma. Is sarcolemma. And within the sarcolemma, better we can understand, here we have cell. So a kind of typical cell. Here this is called as plasma membrane, or we can call it as cell membrane. The sum of plasma membrane or cell membrane. See, when I take it as a cell called as plasma membrane or cell membrane, and when I take myofilament or myofibrils, their plasma membrane refers to sarcolemma. I mean to say the outermost membrane is called as sarcolemma. And when I take cell, so within the cell we will be having the liquid part, is called as cytoplasm. And here I can compare the cytoplasm with the sarcoplasm. Simple word. In cell, the cytoplasm, in case of myofilament or myofibrils, we can call it as a sarcoplasm. Simple word, sarcoplasm. At the same time, in the sarcoplasm or the cytoplasm, we have a cell organelle called as endoplasmic reticulum. Remember? In the cell, we have a cell organelle called as endoplasmic reticulum, and in case of myofibrils, so the endoplasmic reticulum, that is endoplasmic reticulum, are called as sarcoplasmic reticulum. They are called as sarcoplasmic reticulum. Here, sarcolemma refers to plasma membrane or cell membrane. Sarcoplasm refers to cytoplasm. Endoplasmic reticulum refers to sarcoplasmic reticulums or reticulum. Next, see, when I take this myofilament or the myofibrils, so which contain the very important proteins. They contain very important fundamental proteins. So those proteins are nothing but, one will be actin, another one is myosin. Remember. Yes. So that protein is made up of. I mean to say, in case of this muscle bundle. So here, in case of muscle bundle, you will be having collagenous material. I mean to say, two muscle bundles are held together by the collagenous material. And collagenous nothing but it is a kind of protein, no doubt. And here, in case of myofilament, the protein is present. The protein is considered as actin and myosin. So these two are the very important fundamental proteins present in the myofibrils. And this actin is also called as, very confusing words, light band. Yes, actin refers to light band, whereas myosin is called as dark band. Yes, most confusing words. Actin is also called as light band and myosin is called as dark band. And light band is also called as I band and it is also called as A band. Yes, confusion because actin A. Yes or no? But it is a confusing word, so that actin is considered as I band and myosin is considered as dark band or A band. And the actin, see here, this actin or the I band, which is made up of isotropic protein. Yes, isotropic, and the dark band or the myosin is made up of anisotropic. Do remember, isotropic and anisotropic, the bands. The people, they are the kind of proteins present in the myofilament or the myofibrils. This is what a kind of arrangement. Next, see, when we go in depth into the myofibrils or the muscle. So what kind of arrangement? I mean, in case of myofilament, how the actin and myosins are going to arrange. Here we have one unit. That is, imagine we have a long length. That is one muscle bundle. Yes, in the muscle bundle we will be having many myofibrils, structural units. In that, I will take one small unit is considered as here one small unit. Here we will be having So these are the what? One small structural unit, that is one muscle unit. Here we will be having actin and myosin. They are made up of actin and myosin. See, here the actin are going to very near to this H zone. This is considered as H and M zone. Remember, so this is nothing but M line, no doubt. But here the gap between these two, here the gap, I mean to say I can write like this. This gap is nothing but H zone or M zone, Z zone. We have many. Yes, it's considered as the zone. Here, this is normal condition. I mean to say, when we use the word muscles means they have an ability to contract and relax. Yes or no? So they have an ability to contract. Say for example, it's a normal muscle. When I do like this, it undergo contraction. Yes or no? It is contracting. Here the muscles are undergoing contraction. When I do like this, it is undergoing relaxation. See how much? The maximum relaxation. Yes or no? Now the whatever diagrammatic representation I am going to show you people, this is the relaxed condition of the muscle. Yes or no? See, when it undergoes contraction and relaxation, what happens exactly? See, this is normal. We have a gap here. Yes or no? Same gap will be present at the rest condition. When the same muscle undergo contraction, resulting in the, the same diagrammatic representation shifts to like this. Yes, can you understand this point? I mean to say, see, when the muscle is at rest, yes, and when it starts contracting, see, contracting means here the actin and myosin start contracting, resulting in this gap can be reduced. Yes or no? Can you see this point? Here there is a gap and here there is no gap. I mean to say the muscle fibers are going to contract. There is actin and filaments are going to contract each other. And same time here we will be having gap. Can you see this? Yes, at the same time here the gap has been reduced. Yes or no? The gap has been reduced. So means it is contracting. The same time, if it undergo maximum contraction, see, can you see this? So maximum contraction resulting in the diagram is going to shift to like this. Yes, see what has happened here. Here there is a little bit gap between two actin filaments, and here again there is a gap. But when it undergoes contraction, that is minimum contraction. So in case of minimum contraction, that there is no gap here. Yes or no? Very little or negligible amount of gap is there. The same time here also the gap has been reduced because undergoing contraction. The same diagrammatic representation or the same muscle undergo maximum contraction. Yes, maximum contraction. So now the diagram here, these two are where there was a gap. Now these two can be overlapped each other. Yes, sir, can you see this? Overlapped each other, and here there is no gap at all. I mean to say this can be touched to H lines here. This is what the contraction. The same time, when you start relaxing, the same diagram shift to this one, and again relaxation. This is considered as again at rest. So this is what the muscle or the arrangement of muscles within the body. Yes, sir. And based on this, a person called as H. Huxley. Remember the word H. Huxley, a scientist who can propose a theory based on this one called as sliding filament theory. So very simple concept, sliding filament theory based on this whole concept. In that, before understanding the sliding filament theory, we need to understand how the actin is present and it is made up of which kind of protein, where the head is present, where the actin binding site, where the myosin binding site. So we need to understand all those things regarding the actin and myosin. So this is what simple thing. Here we will be having some gap. They are called as zone. Here we will be having H line, M line. Yes or no? Here we will be having zones. So that can be discussed. I mean the sliding filament theory can be discussed in next class. Thank you very much.